Next speaker is Felix. Next speaker is Felix Bärenz von Sanofi in Germany. I would dare to say that you are as experienced with the SURFE² 96 SE as my colleagues are, because you've been using it for quite a while now. So Felix is lab head in vitro biology since 2016 at Sanofi. He received his doctorate of natural sciences from the Heidelberg University and continued with postdoc studies at the University of Ruprecht-Karls, the Ruprecht-Karls University, and then continued to the DKFZ, German Cancer Research Center. So with that, I hand over to you and look very much forward to your talk.
So, hello everyone. Thanks for this very nice introduction. I just want to briefly mention that I think Sanofi Frankfurt is a Nanion client for more than 20 years. And I think we are well experienced with all the systems, especially the SURFE² 96 SE. And today I'm going to show you a few data where we applied SSM-based electrophysiology using the SURFE² technology. And before I will give you a brief introduction where I am coming from. So I'm a lab head in the integrated drug discovery department within Sanofi. So what we are doing, we are developing high throughput assays and profiling assays for small molecule compounds. This requires what we actually need are assays which can be applied in a high throughput manner. This is basically what we are doing. So just talking briefly about the agenda. So I will give you a brief introduction into the system, but I think all the speakers before me already gave a very detailed introduction, so I don't think I need to deep dive into the technology again. Then I will present you 3 assays which we developed for the IMI Resolute Consortium. So we are a partner, IMI partner. And this consortium actually has the idea to develop assays for all SLCs encoded by the human genome, so quite ambitious, and also to characterize novel or unknown SLCs, and also to determine which substrates they are using. So it's a really big consortium consisting of people from academia as well as big pharma. And we were developing, as I said, 3 different assays. Today I'm going to briefly talking about the CLUT9 assay, which we developed, the EAAT family assay, and last but not least, the SLC6 family assay approach. And for all the 3 different targets, we were using and applying SSM-based electrophysiology. I think this slide, I think I can basically skip, because all the speakers before me nicely introduced the technology. And if you don't have any specific question right now, I think I will briefly jump over this. But this is something which was mentioned previously, that you need to generate membrane liposomes in a very good QC, because you want to have stable and robust signals over a long period of time, and this requires a huge purity of your membrane fraction as well as proteoliposomes. And also the coating of your sensor plate is quite essential in how we do it. And we recently published a protocol for SSM-based electrophysiology in the drug discovery background, and you can find it online in current protocols. It's also a cover story. And we are basically using the SURFE² 96 SE from Nanion, where you have 96 different sensors on one plate, allowing a much higher throughput compared to the other system. And as I mentioned, it's a very established technology for us, especially for SLC transporter research. As a brief introduction, so SLCs, I think this was mentioned also previously, are proteins located in the cellular membrane, and they have a really critical role in maintaining cellular homeostasis, and also responsible for drug absorption. And we know from literature that genetic variations of SLCs are associated with several human diseases. And as I said, there are many SLCs in the human genome which are completely not characterized. Nobody knows the substrate. And there are for sure, or there might be monogenic diseases linked to the one or the other uncharacterized SLC. And we can measure those electrogenic SLCs with the SURFE² technology. On the right-hand side, I think the colleague from the previous talk was mentioning this also. When you're thinking about a drug discovery campaign, everything starts with a high throughput screening where you are screening millions of compounds. And I think you can't do it with a SURFE²R. I mean, it's almost impossible to screen a library in a rather short period of time. So you always start with a high content screening, applying, for example, membrane potential assays like the FLIPR assay. Then you go for a confirmation and a counter screen. And later on in the screening cascade, if you in the selectivity phase, you can find the SURFE² as a tertiary or secondary profiling assay for compounds. And we have also established in our labs a high content mass spectrometry based screening system, the ECHO-MS system, for assessing SLC function and transport. I also want to briefly mention that not all SLCs do basically the same. Some SLCs are co-transporters, exchanges, or just passive transporters. And only the electrogenic SLCs can be assessed using SSM-based electrophysiology. And in this graph here, you can see how big the family is and how many SLCs are encoded by the human genome. It's really fascinating. You can see there are 66 subfamilies and 5 non-canonical subfamilies. So it's a huge family, and most of them are not well characterized. So that's for the introduction. Let's now move on with the first assay example. The first assay example I'm going to present you today is the target is SLC2A9, also published as GLUT9. And our aim was to establish a tertiary or secondary assay for drug discovery together with Axam. Axam, as a partner of Resolute, developed a membrane potential assay for GLUT9 for high throughput screening, and we developed the SURFE² assay. SLC2A9 is basically expressed in the apical and basolateral membrane of the kidney. There are 2 isoforms, and it transports uric acid or urate. And it has an essential function in uric acid transport in the kidney and linked to several diseases. So it's a very interesting drug target. And we started to establish a SURFE² assay for this one. Okay. So this slide here is already a sub-figure of our publication, which we submitted a few weeks back. So in this case, we were using HEC293 derived membrane fractions. And we were using HEC wild-type cells as a reference or control, and HEC293 cells expressing GLUT9 isoform 1 and 2 as our SLC preparation membrane fraction, where we want to see currents in the presence of uric acid as a substrate. And this is what you basically can see here. On the left side, you see the HEC923 cells untransfected, where we do not see any currents occurring in the absence of uric acid as a substrate. And as soon as we perfuse in the substrate uric acid, you see an negative charge transfer or negative currents as expected. And this is not only true for isoform 1, but also isoform 2. And you might ask, okay, GLUT9 was published to transport uric acid, but also orotic acid. What happens if you apply orotic acid? I don't show the data today, but interestingly, applying this substrate, we observe heavy artifact currents already in the mock transfected HEC293 cells, which actually happens from time to time. And then you need to optimize your assay conditions in a way to get rid of this basal currents, which you then observe. But I can say as a summary here that both isoforms show specific orotic acid-dependent currents, which is pretty good and a very good starting point for a assay development. Here on this slide, you can see how we continue with assay development. So we test protein dependency, pH dependency. We also do Km measurements to determine the half maximum speed of the transporter. And we test over 10 to 12 different plates how stable the assay is, including a so-called low control and a high control. So a low control is normally a tool compound which we use to inhibit the transport, and the high control reflects basically the normal transport rate of the SLC of interest. And you can see that we have a very nice split between low and high control, and a very good SB value, which is also a prerequisite for a stable assay, which we then later want to apply in a drug discovery campaign. Yeah, and this assay was successfully established in our lab and is now ready for compound profiling in the 96 well plate format, which is pretty good and pretty cool. And the last thing we did for the publication and also for the Resolute to show that we can measure tool compounds or reference compounds in dose response. And this you can see in this crowdy slide here. So we were testing a few urate-1 inhibitors, because if you go into the literature, you can find that some urate-1 inhibitors, so also well-marketed drugs, also seem to have an additional role in inhibiting the transport of GLUT9. So they are mostly non-selective. But interestingly, if you look on the IC50 curves here, when we are using the SURFE² assay, only the very right-hand compound lesinurad showed a kind of dose response behavior, at least in the 3 or 4 highest concentrations applied here. Yeah, and this showed us that we can use this assay as a profiling assay for compounds. And now let's move on to the second example. So also for Resolute, we were developing an SLC6 family SURFE² assay, especially for SLC6 A19, A14, and A15. Again, a brief introduction. So this family is actually, this SLC6 family is a secondary active sodium-dependent transport family for neutral amino acids, and this is actually true for A19, A15, and A14. They are expressed, A19 especially expressed in the small intestine and the kidney, A15 especially in the brain, and A14 in the lung and the gastrointestinal region. And there are also quite a few mutations published for A19 Hartnup disease. A15 has an increased risk of major depression if it's mutated in the brain, and A14, it's linked to obesity if mutated. And yeah, as I said, they have a quite large tissue distribution, some in the kidney, brain, and small intestine. And the first thing actually we did, we again tested our membranes, which were from HEC923 cells, to see whether we can see or get any currents. And here you can see our first results which we obtained. So for 6A19, so 6A19 we published, I think, 3 or 4 years ago already in SLS Discovery. So if you check out online, you will find our SLC 6A19 SURFE² story already online. Then on top, we tested 6A15 and A14. And for all of them, you can see that we are able to generate currents applying leucine, so a neutral amino acid, which was published previously to be transported by all 3 of them. And we can generate positive currents for all of them as expected and published previously, which is pretty good. Then we continued with assay development, and as we have done it for the GLUT9 story, we first optimized the membrane concentration for all 3 different. We tested sodium dependency and also the DMSO tolerance. But because if you are doing drug discovery, most compounds are dissolved in DMSO, so the assay should tolerate 1% DMSO concentration. And last but not least, we also did an KM determination for all the 3 different SLCs, and obtained KM values which are really much in line with already published data applying other technologies. Yeah, and the last thing also for this family, we tested a few tool compounds. In this case, we tested an internal library or compound from an internal library and applied or tested those on all the 3 different SLCs. And as you can see that most of the compounds from our series specifically inhibited SLC 6 and 19 and not the other 2 tested. In this case, we can conclude that we have selective 6A19 compounds in hand, and we were able to confirm this applying SURFE² assay. Before I come to the discussion, I briefly want to mention the last family of SLCs where we did a SURFE² assay development, which is the SLC1A family, also known as the EAAT family. And again, as for the other 2, a brief introduction here. So SLC1A family members are glutamate transporters and are a family of neurotransmitter transport proteins that move glutamate. They are especially expressed in the central nervous system and in the brain, and mostly in the synaptic cleft and in glia cells surrounding nerve cells. And yeah, and this family is also linked to several diseases. So I think still a good drug target, and still a few labs are interested in this family. So here again, as for all the others, we tested the EAT family members using HEC293 cell-derived membrane vesicles, and for all the 3, we were able to generate positive currents in the SURFE² assay. And as for all the others, we tested all the parameters to optimize the assay, checking the right concentration of the membrane per sensor, checking KM value and DMSO tolerance. And again, a quite successful assay development for Resolute. And I briefly want to mention that there is an online database online database available from Resolute, where you can find for most of the SLCs wild type cells, overexpressing cells, and KO cells. And I think you can order those via the Resolute web page. And you can also find in future protocols for all the different assays. You can request the protocols online, and some of the assays were already published. And there's also a nice review on SLC drug discovery available from Resolute. If you go to NCBI, you will immediately find it. So if you are interested in SLC research, I think you should go to the web page because it will be public available, and you will also find all the protocols for all the different assays. It's not only SURFE²R, a huge portfolio of different assays for SLC research, and you can find them online. All right. Yeah, I think I can skip it because here we also tested a few legacy compounds for the EATs, and we're able to show selective inhibition of one or the other EAT family member. And with this, I will come to the end and just present you a brief summary and outlook. So we did a successful assay development for SLC2A9, which is also known as GLUT9, SLC6 family, and SLC1A1 family, applying SURFE² technology. We tested successfully tool compounds from literature and internal library and validated the 3 different assays. And these assays are now ready for compound profiling. I think the SURFE² pipeline in Sanofi Frankfurt is an important pipeline for drug discovery. And we validated this technology for transporter screening trees, a secondary profiling platform. And the GLUT9 story was already submitted, as I mentioned previously, for publication in collaboration with Exam. Last but not least, a few acknowledgements. So all the assays were not done by myself. They were actually done by Antje, Katerina, Yasmin, and Jennifer in the lab. And I would like to thank all of them for their excellent lab work and entire data evaluation. I want to thank Exam for this very nice collaboration. Thomas, who is in the audience here, for scientific input and critical review of data. And also IMI Resolute for providing all the different cell lines and membrane batches we obtained for the assays. And with this, I would like to thank you for your participation and listening to my talk. And I'm willing to answer a few questions.
Thank you very much, Felix. Thank you. Questions? I have one. So when you have a new transporter target, in the initial phases of establishing the assay, what would you say are the major challenges?
I think the major challenges is to get a good membrane fraction. So when you do membrane fraction preparation, you need a good membrane fraction. You need a good expression of your transporter of interest, because we found out if the transporter of interest is not expressed in a very high fashion, sometimes we have problems to observe currents or specific currents. This, I think, is essential. Yeah, and also you should know which substrate is transported, because sometimes it's not known which substrate is the best or the relevant substrate of the transporter, and then you have to play around with the transporter, with the substrate.